been talking about for the past several weeks. And maybe I should sort of warn you that it's kind of ongoing work, and it's been ongoing since about 2007, and I wouldn't claim to have actually understood anything as yet, but at least I'll attempt to describe what we're trying to do. So this is a kind of collaboration with my kind of long-suffering colleague Andrew Free, who's a microbial ecologist, also in Edinburgh, and we kind of got interested in this back in 2007, and we thought it was a beautiful model system, and it would be really nice to study it. And little did we know how much trauma it would cause us over the intervening years, but we're still here. We're still working on it. And so the actual work that I'll present has been done by various. Eulin was a postdoc in Andrew's lab, working with both of us. Fiona's a lab technician who works 1 day a week, and Helen's another lab technician. Tim is my student in physics, and he's a sort of a theoretician, but now he's got interested in doing experiments. And Ian is our kind of saviour from geosciences, who knows all about how to measure sulfide gradients, it turns out, and that turns out to be important. So what are we trying to do? I guess what we were sort of inspired by back in 2007 was the idea that it would be nice to have a model system where you could really try and interrogate this one system and understand what's going on in microbial communities, because there are all these complicated things going on. So you have metabolic interactions and viruses and the question of how you should even model the population dynamics of the microbes, because you have noise and spatial structure. And so no one really knows what controls, out of all these factors, what controls the community structure and function. And we thought that it would be nice to study a system where you can really have a lab system that you can interrogate in a well-controlled way, and you can make replicates, and you can also perturb the system in well-controlled ways. And so that was our motivation. And so what we thought that the model system should be controllable. So you should sort of be in control of the thing rather than it being in control of you. It should be perturbable, easy to sample, but it should also be realistic. So that the thing should actually be somewhat like a real system. But it should be.
Not all think these things completely contradict.
Well, maybe. But it should be simple enough that you can sort of understand. And in principle, it should be modelable. And so the last part of my talk, I'm going to talk about our attempts to model this thing. And I'm not sure it really is modelable, but we'll see. So this is the system that we sort of decided to study, which is the Winogradsky column. And this system has been around since the late 19th century, when it was invented by Sergei Winogradsky, who was a Russian. And basically, here's what one of ours looks like. So it's a plastic tube, and in this plastic tube you put pond sediment and water from a local pond, which in our case is Blackford Pond in Edinburgh, and you supplement this with a carbon source, which is cellulose in our case, and a sulfur source, which is calcium sulfate. In our case, what we do is actually sterilize the system at that point, and then we inoculate it with a small sample of a microbial community from either the same or a different source, and I'll explain why we do that later. But in any case, what you have is this system of sediment and water, and you then incubate it for several months in an environment where there's light. And what happens is a spatially structured microbial community develops, which is nutrient cycling, and it's stable for actually years. So this system, without opening it, it just maintains itself, and it does nutrient cycling. And what we like about it is that you can track the chemical gradients. So by sticking electrodes into it, you can measure the vertical profiles of oxygen and sulfide and other things too, if you want. And you can also sample it to find out the species composition. So we sort of thought that this is a nice system because it's in the lab, and you can make multiple replicates, and you can perturb it in different ways, and also you can sample it. And it's got this spatial structure which we kind of liked.
How big is that?
Well, this particular one is 15 mils, so it's sort of this high, but we make ones of different sizes also. So what goes on in this system? Well, here's what we think goes on. Definitely you have a gradient from anaerobic at the bottom to aerobic at the top. So the oxygen goes like this. And then at the bottom you've got sulfide, and so you've got these kind of opposing gradients of oxygen and sulfide, which is a bit like what you get in the ocean. So it's a sort of a kind of model system for gradients that set themselves up in real sediment water bodies. And basically, in our understanding, there's kind of two cycles that go on, and one of them is the carbon cycle. So you have photosynthesizers that fix carbon, and then they die, and they get biodegraded by guys at the bottom. And so there's a carbon cycle, and there's also a sulfur cycle between sulfate at the top and sulfide at the bottom, basically. And so it's a sort of a model for nutrient cycling more generally. And it's got lots and lots of microbes in it. So one day we made an attempt to estimate how many microbes would be in one of these things, and we thought that it was probably 10 to the 8 to 10 to the 9 or even more. So it's a kind of a complex community. And what we also, I'll show later, but when you do sequencing on this thing, you find out that it's got thousands and thousands of different species, as you would expect. So it's a rather complex community. So what are we doing? So I thought I put in this slide just to give a kind of overview of all the things we're trying to do with these things, with these Winogradsky columns, but I'm not actually going to talk about all these things today, mostly because none of them are finished. But okay, so what we're trying to do at the moment is understand how this thing develops in time. So here's a little bit of data to illustrate what kind of thing we're doing there. So here we have a whole collection of Winogradsky columns which we sample once per week. Well, here we didn't sample them, but we actually measured them with electrodes. So what's plotted here is the total amount of sulfide at a certain depth in the sediment as a function of time for columns where we put in different amounts of carbon, so different amounts of cellulose. And what you see is you get this kind of interesting peak in the sulfide, which changes depending on the amount of cellulose you put in. And at the same time, we can also take samples and do DNA sequencing on them. And so this is the exact same experiment, except what's plotted here is a sort of map of the community composition. So each of these dots is one of our samples, and the colour tells you which cellulose concentration is it. So the blue ones are the high cellulose, and the red ones are the low cellulose. This is the starting sediment. And it doesn't really matter what's plotted here, but what you can see is the community changes in time. So where it is on here tells you sort of what it's like in some sense. And you can see that we can really see these trajectories of community composition. And one day we would like to have a model that sort of links this, that links this to this and explains everything. And currently we don't have such a thing, unfortunately. So that's one thing that we're trying to do. We're also looking at what I'll maybe talk about later, how the system responds to nutrient perturbation. So as you change, for example, the amount of cellulose or the amount of sulfate, what happens? So in particular, we're interested in whether the community responds in a sort of gradual way or whether it kind of undergoes a catastrophic collapse at a certain threshold amount of cellulose, for example, which is a kind of model for organic pollution of water bodies.
But so the cellulose, so are you adding it to the bottom or to the top?
We just homogenize the whole thing at the start.
So it's all homogeneous.
Yeah, but it's basically at the bottom because it sinks.
So, and that's like how Winogradsky set it up. Like he put cellulose on the bottom, but that doesn't match so well because the cellulose should be landing on the top of the system, right?
Yeah, but I think it probably sinks in real life as well. Yeah, what we do is we take a big pot of sediment and we sieve it to remove kind of sticks and stones.
Yeah.
And then we add the cellulose and the sulfate and mash the whole thing up, and then we aliquot it into the plastic tube. So you move as certain as you can be that they're all the same, basically.
And then that sequencing, is that metagenomically or how it?
It's 16S currently.
Okay.
Yeah. So another thing that we've been trying to do since about 2008 is actually make many replicates of these and find out whether they actually come out the same or not. And we're still working on that. Something that's quite intriguing is how this thing depends on the system size. So what we find out is that small ones are more variable than big ones, which every statistical physicist will tell you should be the case. But when you actually try and think about what's really going on, it's not quite as simple. So we're still thinking about that. So that's a sort of overview of what we're trying to do. But one thing we have actually sort of finished, which is the only thing that we've kind of finished, so I thought I would tell you about that. And that's this project about predictability. So we managed to publish this thing last year in ISME Journal. And the question that we're trying to ask here is really, can you think about this system as a kind of collection of microbial pioneers that are colonizing this pristine environment because we sterilized the microcosm, so it's kind of pristine, and then you add this inoculum of pioneers, and the question is like, what happens and how predictable is the outcome? And in particular, if you do an experiment where you, for example, do the exact same set of pioneers, 10 times, would you end up after 2 months in our case with different communities, which would be sort of equivalent to this, or would they somehow all go down the same trajectory? And conversely, if you started off with different sets of pioneers, so different inocula, would you end up all converging to the same community in the same environment or not? And that's what we tried to test with these columns. And so we were sort of inspired by, colonization of a pristine new baby's gut and other such problems. So what that translates to in terms of our experiments is kind of this. So if we're thinking about this case where you have the same set of pioneers, then basically what you want to do is inoculate identical Winogradsky columns with the exact same starting sediment and see whether after 2 months of development they come out the same or different. That's the first experiment. And the second experiment is if you inoculate the same Winogradsky column but with different sources, are they all going to end up being the same? And so that's the question we were asking. And so the way we did that is to sample sediments from all different lochs in Scotland. So we kind of went round with a boat and sampled these sediments, and we actually sampled 7 different water bodies in Scotland, including one which was the source sediment. And so then we had these 7 samples, and they were our 7 different inocula, and each one of those we used to inoculate 3 different replicas. So by comparing what happens between the inocula, it's like we're testing whether they all converge to the same community. But then by comparing what happens between identical replicas from the same inocula, we're testing whether they diverge or not. So that's the plan. And what we did is we took these sediments and then we incubated the columns at 25 degrees C for 16 weeks, and then we sampled them.
So can you tell which loch the water comes from?
You can tell if you put a label on the online it's saying. What you mean if you just chemically sampled it? I don't know if you could tell which loch it came from, but there are differences in the pH and other things, and there's definitely differences in the source community within one loch. No, not entirely. But what we did is we just took one core and then kind of mashed it up, so we could be as sure as we could that the inocula were really the same, as far as you can make them the same. But it is true, at least with Blackford Pond, we've tried sampling it in different places and different depths of, how big the core is, and it comes out different, so you have to be a bit careful with that.
And the water is all from one spot?
No, so the way it works is the microcosm is always from Blackford Pond.
Yes.
So, and then it gets sterilised. So the environment is always the same, and when you put in a very small inoculum, which is just sediment, so there isn't any pond water coming from all these different places. It's just sediment. And that's the source of the microbes. The water always comes from Blackford Pond and it always gets sterilised. Does that make sense?
Do you mix the added sediments, or do you just put it on top, or how does it work?
You put it in and then you shake the thing up.
Okay.
Yeah.
And how much are you adding?
100 microlitres
So
of the sediment.
Of the inocula.
Yes. So it's kind of a small amount compared to the size of
So
the...
you've only got, what, 10 to the 7 cells?
Um,
Because you'll have about
probably
28 per gram.
yeah.
28 to-
So 10 to 6, 10 to 7, I would guess. Yeah. Okay, so then after these 16 weeks, what we measured, in this case, we didn't actually measure sulfide gradients and so on because we did this quite a long time ago and we didn't know how to do that then, but now we do. But instead, we actually just measured the redox potential between the top and the bottom, which is a kind of measure of community function in some sense. And the point is that what we saw is that the replicas between triplicates from the same source didn't come out the same, and also between different sources they didn't come out the same. So what this kind of tells you is that in terms of function, somehow these microbial communities are not all identical, and there's some variability between replicates. But maybe what's maybe more interesting is actually looking at the community composition, so like which microbes are there. And actually, so this is a bit embarrassing because we did it quite a long time ago. So we were using this technique, which is now outdated, which is called DGGE. And in this technique, what you do is you take your sediment sample and you extract DNA from it, and then you amplify up this fragment of the 16S ribosomal RNA gene, which all bacteria have. And so you have a whole collection of fragments of the same length but different sequences. And then what you do is you run them on this gel, which has a gradient of denaturing, and so different sequences stop at different places. And so each of these lanes here is one sample, and the position of the bands on the gel is telling you which species you've got in the microcosm. So it's like a fingerprint of your community. But unfortunately, you don't actually know which band corresponds to which species, so it's a sort of a fingerprint without any labels on it. But what you can do is at least quantitatively measure the similarity between different communities by, in fact, what we use is something called the Bray-Curtis index, but it's a measure of how similar two fingerprints are, and it takes into account the intensities of these bands. So we did that, and then what you can do is you get this matrix of similarity. So sample 1 has a similarity with sample 2 of whatever, 0.5 or something, and you can plot a plot like this. So this is a two-dimensional graph, which actually is the thing that I showed before. But the point of this plot is it's an attempt to project the similarity matrix onto 2 dimensions, such that the distance between any 2 samples on this plot is somehow related to their dissimilarity. So samples that are all similar come out in a cluster, and samples that are different from each other will come out with a bigger distance. And the point of this is you can sort of look for clusters on such a plot. I guess most of you knew this before, but some people maybe didn't. Okay, so the first question is this one. Like, if you just start off with these exact same pioneers, then are they going to come out the same after 16 weeks? And the answer is no. And you can sort of see that in these gels. So this is a gel of the archaeal community from Loch Leven. So that means the microcosms that were seeded with the community from Loch Leven. And here you have the initial sediment, which we ran on a gel, and next to it you have 3 microcosms, which are supposedly identical. What you can immediately see is that there are differences between their final community compositions. And that's also true from this other set. This one's from Loch Lubnaig. And more recently, we've managed to do some proper sequencing analysis with 16S Illumina sequencing. And to our great relief, we see the same thing. So here is some sequence analysis at the genus level of this same experiment. So this is also Loch Lubnaig. And again, you can see that these 3 replicates didn't come out the same. So that's good.
You haven't falsified it. There doesn't need to be any relationship between this and that for that to be true. But yes.
Well, it would have been a bit worrying if these had all been identical, because yeah, luckily they weren't.
It doesn't mean that's meaningful.
No, but at least they're both equally non-meaningful. But okay, so the answer is that there is a divergence between these replicas in our experiments.
Because you see already from the gels, so
Yeah.
mathematics is not really necessary. We have to show this.
Well, it's not mathematics, but it's just you get more information from this technique, and also people believe it more. So we even believe it more. So
And you can share it with other people.
yeah.
But in a sense, it's just a translation of this figure into another figure.
No.
It's another type of visualization.
No, it's not just visualization, it's also what you know about the sequence. So in this technique, what you do is you take your collection of these DNA fragments and you post it off to a sequencer, and what they send you back is an enormous spreadsheet of the abundances of each sequence. So you actually know which species each one is to some extent. So it's more information, actually. So the conclusion of this is that the replicates come out different. But when you make one of these 2D plots, so here the color coding is telling you what the source community was of all of these microcosms. What you actually see is they also cluster according to color, which is telling you that even though they diverged between replicates, they still remember where they came from, because they're somehow clustering. And you can actually do a statistical analysis of like how much of the variability comes from the source compared to random divergence, and it's about 50-50 according to the statistics. So it seems like there's a contribution from the source, but there's also a contribution from this kind of variability that we don't really know where it comes from. Just briefly, the other question that we mentioned is if you have different communities, do they all converge on the same final community? And the answer to that's clearly no. And you can also see it in this plot. So in this plot, it's the same as the other one, except now we also include the source communities, which are the ones with the circles around them. And what you can see is that actually our source communities were more similar to each other than the final communities were to each other. So rather than having a convergence, we've actually got a sort of divergence going on among these different sources. And you can test that statistically as well. So that's what's happening. Another thing you can ask is if you just look at an individual taxon, what happens to it? So is it true that the final community is a bit like the initial community or not? So if an individual taxon was abundant at the start, is it still abundant at the end or not? And so you can do that by making a plot like this. So this is the abundance of taxa at the start in the sediment that we inoculated with, and this is the abundance at the end. And you can do that for each individual taxon in your data set. And the answer is that there's apparently no correlation whatsoever. So a taxon, many of the taxa which are actually abundant at the end were undetectable at the start, and vice versa. So what we conclude from that is that there's just this huge rearrangement of the community that happens in our experiment, and so the final community bears no resemblance to the initial one as far as we can tell, which is perhaps not surprising because our experiment is done at 25 degrees C in a lab, whereas in the loch it's kind of much colder than that, and so on. So maybe that's not surprising, but it's sort of interesting to see. And so what we kind of concluded from that is that when this experiment happens, there must be a sort of strong selection for species that were initially rare. So it must be something about the experimental conditions that kind of requires species which you don't have very many of at the start, which you don't have very many individuals of at the start. Does that make sense?
So there is some repeatability in which species rise in frequency. If we put it, there's a set of species whose mean frequency increases a great deal.
I'm not sure that's true. So I don't think it's here. But we also did an analysis where you look at the replicates, the replicates from Loch Lubnaig, for example, and you look at individual taxa across those replicates, and you say, like, do you see reproducible amplification of any particular taxa across the replicates? And the answer is also no. So I don't think we saw any reproducible changes in taxon abundance, really, in our data.
Then there's no natural selection.
Um,
Of course there is natural selection, but...
Well, it could just be different in different replicate microcosms, right? But no, I see what you mean. I see what you mean.
Well, you're sort of saying that the stochastic randomness of which things you happen to have in your small inoculum determine everything.
Determine a lot. They don't determine everything because you have this history dependence, right? So it does remember where it came from. It's not completely stochastic, but 50% of it is determined by something we don't understand, which is somehow random. Maybe I shouldn't have mentioned selection because it's true I don't really understand what I'm talking about with that.
Did you do one like the common garden thing where you just, so you put the inocula in and then just divided them into 3 replicants? Or did you?
That is basic. No, we didn't do that, but I don't think it's any different from what we did do, really.
You should get the same thing then.
Yeah, it's true that we didn't do that.
Okay.
Did you look at the functions actually? So meaning genes?
No, because this was all done with the DGG, but now we have the sequence data, so that's something we're going to do. Yeah, but we haven't done it yet.
But you saw different, phenotypically you saw differences, right? Because
Yes, well, the redox gradient is different. But it could be that in every case you have a population of photosynthesizers and a population of sulfur reducers, and maybe they're just kind of different in abundance. So it would be really nice to see that actually, now we have the data.
Can I ask a question on the timescales in the system? Like, do you have any idea how long it took before the spatial gradients in sulfur and oxygen developed? And how,
Yes, we do know that
you
now.
sampled it?
Not very long is the answer, about a week. You start to get spatial gradients. And then you get this peak in sulfide that I showed at the beginning. This one. So that happens, well, it sort of depends, but here it happens at about 8 weeks. But you have a gradient really early on.
And the endpoint that you sample is after 20 weeks?
In this case, it was 20 weeks, yeah.
But in the other graphs?
16.
16.
Yeah. Yeah, so it's about here. Okay.
So Rosalind, I have, you go back to the last, here. So you have a conclusion that strong selection for initially rare species?
Yeah, well, maybe that's not in, yeah. I'm not sure I should really have said that. No, we kind of hypothesized that. So we were trying to explain why is it that that many of the species or many of the taxa which become abundant here apparently had 0 intensity in the sediment, which just means it was low because the detection threshold isn't very good for DGGE. And so what we hypothesized is that in those individual communities, something must have been happening that kind of favored those initially rare species. Whether they turn out, whether they're actually important or not, we don't know.
Yeah, I see it's a hypothesis, but I don't see there's any evidence from...
No, not necessarily.
Okay.
But there is evidence that some of these guys came from rare to abundant.
That's true.
I think maybe that's all you can say.
Yeah, but that could be a completely stochastic process.
Yeah, it could be. And now that we've got the sequence data, I think we're going to learn a lot more because we're going to find out who those guys were.
Is there any reason why you did this at 25 degrees C?
Well, that's what our plant sciences people
Chips
had
away.
turned on in their
I
plant
see.
sciences
Yeah.
room. But no, it might have been better
Yeah.
to do it. I mean, I don't think you're ever going to really reproduce a loch, so you might as well kind of give up.
It'll
with that.
never get to 25.
No, but anyway, I mean, the loch doesn't have cellulose and sulfate, so I don't think we can pretend that this is a good representation of a Scottish loch. Anyway, it's just a model system.
But I mean, you're perturbing the organisms a lot.
Yeah,
Just by making it whole.
yeah, sure you are.
The second point I want to say that when you look at the chemistry, the profile of sulfide show us
Yeah,
that same group, the chemical evolution is almost identical, right?
yes,
Sulfide over time.
yes.
You remember that, right? Okay, then when you look at
I know what. Okay, but hang on a minute. So that first data set were much bigger microcosms. Maybe I
Okay.
should have said that. So the ones I showed at the beginning with these peak are actually 250 milliliter,
okay,
and these ones here are 15 milliliter,
okay.
and that makes a big difference with the variability.
So what type of variation in the chemical profile?
Yeah,
these?
that I don't know because we didn't measure sulfide gradients. But the only thing we measured were these redox potentials, which are variable. So this is the redox potential.
Okay.
Actually, for small microcosms, it's quite difficult to measure sulfide profiles because they're too thin.
And then for the chemical part, did you do the sequencing on them? Those large size
Yes.
carbon?
Yes.
Are they different or the same?
Good question. I think they're much more similar. Yeah. Well, we've done DGG, and
Yes.
they're by DGG they're indistinguishable.
Indistinguishable.
Yes.
So it means they're
Means
close to
that
each other?
yes, much closer than these.
I see.
But these ones are small, and those ones are big.
It would be interesting to see for the small ones how the chemistry differ.
Yes.
among
Yes.
them.
For that, you need a different shape, I think, because it needs to be fat enough that you can get the electrode.
Because I suspect that the impact of those individual identities on the overall chemistry might be small.
Yeah. Okay. So maybe you get the same chemistry in different species.
Or
That would
put
be
it,
nice to see.
or put it a different way, the diversity of organism on the overall chemistry of system is very minimal.
Yeah. So that would be very nice to see if that's true.
Right.
Actually.
Yeah, it would be lovely, yes.
Yes. Maybe we'll see it. Yes. Okay, so what we kind of concluded for this, maybe you're going to disagree with this, but anyway, what we found out, what we concluded is that when you take these inocular samples, even though you think you've taken the exact same inoculum 3 times, the rare species in it will not be the same because you're just sampling statistically. And so what we hypothesize is that maybe this divergence between communities is simply due to that you have these rare species and you didn't sample them the same, and so they came out different.
So you haven't actually tried to sequence a set of inoculum to see how.
No, we haven't, but I'm sure you wouldn't see anything from that because even in 16S sequencing, you're not going to see much difference between 3 identical samples from the same.
You said there's only 10 to the 7. So how well mixed are they?
And you will miss all the less abundant species.
Yeah,
You
so
missed the
I
very
don't think
rare
you'll
ones,
see
which-
it.
which make the difference.
Yeah,
Yeah, which make the difference in your result.
yeah. So I don't think there'd be any point in doing that, honestly. Okay, so we thought to ourselves, well, what happens if we can manage to use a different inoculum which somehow is pre-adapted to the microcosm environment? So abundant species should stay abundant, and less abundant should stay less abundant, and the thing should be less variable. And the way you can do that is using as an inoculum a mashed up microcosm instead of a sample from a loch, because in principle that system is already acclimatized to the microcosm, and so that's what we did. And so and these are the results. So what you see here is each of these grey dots is one of our replicates from an identical source. So you have 8. In this case, we did 8 replicates from the same source, and they have this divergence that we saw before. But then these little ones here are the daughters of this one, which we did also replicates. And this one here is the daughters of this one. And what you can see is that indeed it's true that the variability is much less when you use an inoculum that came from a kind of pre-adapted community. So, so that's what we did. And we kind of concluded from that in some sense we maybe were right, that maybe it is these rare species which somehow determine this divergence. Another thing we did that I didn't put in the slides is we also did this taxon, start and finish abundance correlation. You do see more correlation in this case than you did before. So to some extent it's true that abundant species stay abundant in this experiment. So that's kind of what we did in that study. I guess we sort of concluded that at least what we can say is if you have a pre-adapted community, it's more predictable. And that could somehow be useful, because if you're trying to design a wastewater treatment plant or something and you want to know what community you're going to wind up with, the message from our study is you better start off with one that came from a wastewater treatment plant and not from somewhere else, which maybe is obvious. But so this study leaves many questions, and it was a bit frustrating in that sense. And I guess I'm a kind of theoretician by background, and so for me the most frustrating thing is that we don't have any sensible model of what's really going on in terms of the dynamics of metabolic types and species in these communities. And so in the remaining time I'm going to describe our attempts to develop such a model. Okay, that's what I'm going to do. And that's been done by my student Tim Bush. And so the first thing to say, well, that first part of the talk really focused on diversity. So, how many species you've got and how they diverge. And so it would be really nice to make a model that includes species diversity. But what kind of thinking about that, we sort of realized that we don't know how to do that. And actually, the first thing we should try and do is make a model that describes how different metabolic types change in time, because at least then you've got a chance of linking the chemical data with some kind of model of the populations. So that's what we tried to do. So what we said is, let's pretend that this is what's going on in our microcosm. So it's a sort of highly simplified picture of what we think might be going on. So you have light, and the photosynthesizers are a microbial population that uses light and produces oxygen. And then that oxygen gets used by sulfur oxidizers in the sulfur cycle. So it gets used to turn sulfide into sulfate. And then coming from the other side, you've got the cellulose that you put in, and that gets degraded by the cellulose degraders, and they produce hydrogen and also acetate and other things, but we just pretend it's just hydrogen for now. And that gets used by sulfur reducers to complete the sulfur cycle. So in our minds, this whole system can be kind of simplified into a cycle of sulfur, which is driven by inputs from both sides. So you have an input of hydrogen or whatever other electron donor, and another input of oxygen. So we're just going to pretend that that's what's going on in our system and do the modeling of this and find out what it does. Okay.
Question. Are some of the photosynthesizers also using sulfide, like the green?
No, not in this model, but they should be. So Tim has later done that, and it makes a difference. So okay, but not in this talk. So that's what we decided to do, and of course in reality you have like 200 different species of sulfur oxidizers and 200 different species of photosynthesizers, and it would be nice to know what they're all doing, but that's not in our model. Okay, and then we thought, well, let's make the thing even simpler and let's just focus on the sulfur cycle. So now we're going to have a model which has only got two populations, the oxidizers and the reducers, and we're going to use parameters to mimic the supply of oxygen. So the growth rate of these sulfur oxidizers is going to depend on the oxygen concentration, and the growth rate of the reducers is going to depend on hydrogen concentration. So we have basically what we've got is two cross-feeding populations.
Is there inhibition? The reducers are sensitive to oxygen?
No, not yet, but you could put that in. Okay, so then you can write down some equations for that, and these are the equations that we decided to write down. So what you say is that the growth rate of the oxidizers depends on the amount of sulfide, that's S1, in a saturating way, which is a mono growth function. So if you have too much sulfide, then the growth rate will saturate. But if you have only a small amount of sulfide, it will be linear in the sulfide concentration. Then we put in this term here, which turns out to be important, which says that the population can be limited by something other than the amount of sulfide. So in our case, that might be carbon limitation or limitation by iron or whatever else. And so what this term says is that if the population reaches a size X max, then it will stop growing. And that mimics the fact that you have other things other than sulfide which are necessary for growth in the system. And then we just have this death term. And so then you can write down the same equation for X2 and equations for S1 and S2 and so on and so forth. And it turns out that for the steady state, you can solve this thing analytically. So you can just write down what is the steady state amount of S1, for example, as a function of these parameters V1 and V2, which are the maximum growth rates of the oxidizers and reducers. And the reason we like these parameters is because these are the ones that we think are proportional to the amount of oxygen or the amount of hydrogen. So by varying V1 and V2, we're pretending to vary the amount of oxygen or the amount of hydrogen, so the kind of drivers of this system. Okay, and so what's plotted here is the results for that simple model. And here we have the ratio of V1 over V2, so like how much oxidative versus reductive driving is there. And here we have the fraction of reduced substrate, so the amount of S1 compared to the total of S. So that's a kind of model of the redox state of the whole ecosystem in our minds. And the interesting thing is that under some circumstances, this thing does a switch. So as you vary, for example, the amount of hydrogen, it does nothing, does nothing, does nothing, and all of a sudden it crashes into this oxidized state. And whether or not it does that depends on the total amount of sulfur. So if you have a lot of total sulfur, then it will do it, and if you don't, then it won't. And you can make a sort of colour plot of that if you want. Okay, so that's what happens, and why does it happen? So here's our understanding of why it happens. So in our model, we think that it's like a tug of war, right? So you have the oxidizers that are trying to oxidize the sulfur, and you have the reducers that are trying to reduce it. And so this is a kind of how reduced the system is. But it's a weird kind of tug of war, because the more these guys win, the more food they make for the other guys, right? Because the more reduced the sulfur gets, the more substrate there is for the oxidizers, and vice versa. So under normal circumstances, you wouldn't expect any sort of switch-like response, because the activity of one would just balance the activity, it would just stimulate the activity of the other, and the thing would just respond gradually. But because we put in this term that saturates the growth, so at some point the population reaches its maximum size, and then it can't respond anymore to the amount of sulfur because it needs something else, like carbon or something. Then at some point you can reach a state where one of the populations just can't respond anymore because it's already reached its maximum population size. And in that case, then stimulating one of them will just cause the whole system to collapse into this other state. So what we sort of discovered is that under circumstances where the population can saturate, then you can get this switch-like response of a nutrient cycling ecosystem. And it turns out that maybe, I don't know if you're interested in this, but the maths of this actually maps onto something that's already known in intracellular signaling networks, which is called a zero-order ultrasensitive response. So that's interesting.
The cycle is, for example, phosphorylation and dephosphorylation.
Yes, exactly. Yeah. So in that, you have one enzyme that does phosphorylation of some substrate and another enzyme that dephosphorylates it. And it turns out that the kinetics of that is actually exactly the same as the kinetics of this. And there also you get the switch-like response. So that seems sort of nice. And then, and so we thought we'd simulate a more realistic version, which is this one. It's got 2 boxes for the oxidative and reductive zones. And now it's got 4 populations, so it's actually got photosynthesizers and cellulose degraders and hydrogen and oxygen. And you can sort of write down equations for all of that system and simulate it numerically. And so you can get a plot like this, which is, so now instead of V1 and V2 as our parameters, we've actually got cellulose and light, because those are the things that you would actually vary in a real experiment. But the more light you put in, the more oxygen you put in, so it's kind of like varying the oxygen or the hydrogen. And you can get a sort of colour plot. So where this is red, it means that it's reduced the ecosystem, and where it's purple, it means it's oxidized. And again, so for example here, if you vary the amount of cellulose at fixed line, it predicts that there's going to be this kind of switch-like response. Okay, so that was the sort of prediction of the model.
Can I ask a question?
Yeah.
It's a 2-box system.
Yeah.
So it might be that one part is reduced and the other is
Yes.
oxidized.
Yes.
But in this graph you talked only about.
Yes, I know.
It is.
We maybe should have looked more detail at that actually. So what we did in this analysis is just add up the total amount of sulfide, for example, without distinguishing, which maybe there's more information actually that we should look at. It's true. So Tim, having done this, got sort of inspired to try and reproduce this thing experimentally. And so he set up 156 Winogradsky columns. This time they're slightly bigger, so these are 15 milliliter ones, with different amounts of cellulose in and also different amounts of sulfate. So he has 2 different sulfate concentrations and I don't know how many cellulose concentrations, maybe 15 or something. and he incubated them for 20 weeks, I think. And then this guy, Ian Butler, we met him, and he turns out to be able to measure these profiles of sulfide using voltammetry, which is very useful. And the data you get from that is something like this, so the depth in the microcosm and the concentration of sulfide. And so by doing that, you can actually sort of plot the same thing that we predicted in our model, which is the total amount of sulfide, which you get by integrating these depth profiles as a function of the amount of cellulose that you put in at the start. And so those are the results, and the two lines are for different amounts of sulfate that went in at the start. And so on the plus side, it does sort of do what the model says it's supposed to do, which is that as you increase the cellulose, which is sort of like increasing this parameter V1 over V2, you do indeed get a switch from a non-sulfidic to a sulfidic state. And so at one point we were quite excited about this, but then we talked to various geologists, and they said, oh, you know, of course you see this for various reasons. And the first reason they said is because you have iron in the system, and iron too reacts with sulfide, and to make a kind of inert iron sulfide. And so the first geologist that we talked to said, well, all that's happening here is you are producing sulfide, but it's reacting with the iron, and so you've got a kind of iron buffering effect going on. And so this is where the iron buffering runs out, and you start seeing this. And so, but we don't think that is what's going on, because we actually measured the concentration of iron after that, and it's too low to explain this. So we don't think it's that, but it could be some other stuff. And so Tim actually is now trying to make more detailed sort of geochemical models, actually using some software that Chuxing has been involved in developing. And we kind of, we don't know the answer yet, but we think that maybe you don't need this population saturation effect to explain this. It could be explainable in models that don't have that. So we're still struggling with this, basically. Yeah.
I'm curious, how much iron is too much?
I can't remember, to be honest. But what you have to do is work out how much sulfide you made. So you know, how does that go again?
How rich in iron your systems are?
Not particularly, I think. I mean, it's,
In freshwater lakes or even water system, iron is very small.
yeah. I forget the exact number.
Unless
But it was low.
In the sediments, you have a chance of higher iron. But in lake sediments, pretty little.
Yeah.
But
So-
this profile, I think I have a simple way to explain that.
Oh, good.
If well, maybe not. Maybe my explanation is wrong. That's why when we add a cell, so first of all, this profile, they show the state concentration doesn't tell you the flux, it's just a state parameter, doesn't tell you the flux. So that's number one. Number two, as you add the cellulose, you provide more electrons.
Yeah.
And electrons doesn't have, so where will electron go? That I think goes to the photosynthesis, that take up the electrons, produce oxygen. And this photosynthesis are always limited by the sunlight. So as you add the cellulose, you are approaching the maximum capability of photosynthesis, or even you go beyond that. And then when you go beyond that, those sulfide were waiting for the photosynthesis to produce oxygen to get rid of it.
Yeah.
So sort of you are.
I think that's what Tim thinks. So he's got a model where you've got a kind of inflow of oxygen. He hasn't his model so far, the one that he's doing in the spatial version, doesn't have photosynthesis, but has a sort of oxygen inflow rate from the top.
Right.
And I think that's what he's saying basically, actually.
So you have, basically you have two competing processes,
Yes.
one wins over the other.
Yeah.
That then determines whether the sulfide goes up or down.
So I think that's what he thinks too.
Okay.
But we should talk in more detail. So also another funny thing about this is it sort of looks like there's two steps, which may or may not be true, but okay. So that's it basically. So what we're trying to do now is make more realistic models, as I said, with proper spatial resolution and proper chemistry, and maybe also different growth rate functions, because this whole business of the saturation of the growth rate is a little bit kind of ad hoc, and that there might be different ways of doing that. And so the question is really, can we reproduce the dynamics? Not only can we reproduce Tim's switch thing, but also can we reproduce this dynamics that we saw where we had this sulfide peak in the ones that we sampled every week. And then we've got this huge mass of 16S data that showed up a few months ago, And so we're really trying to analyse this and find out stuff about which species is it which really are responsible for the variability and stuff like that. So that's kind of it, I think. So longer term, I think it would still, I would really like to make models that have got species diversity in them, but I just don't know how to do that, unfortunately, at present. And also, I think it'd be really nice if you could make a Winogradzki column which is synthetic, in the sense that you know which species you put in. So if you could do that, then you could really control the diversity, and you could ask questions like, you know, how much does it matter whether you've got 5 photosynthesizers instead of just 1, and stuff like that. So I think that'd be really nice, but we haven't done it as yet. So that's the end, I think. Thanks.
Do you have plans to look at the viral fraction?
It would be very nice to do that.
Because it seems like you should have, especially, I mean, a lot of your differences could be because of selective pressure by the viruses that you're adding in.
Yes, but how? I mean, I think I know how to count viruses. At least I know someone who counts viruses, but I don't really know how to assess their sequence diversity.
If you,
Maybe you do.
I mean, yeah, but it seems like if you do instead of the 16S,
Yeah,
you're sequencing a total, you know.
some phage protein. You think?
No, I wouldn't pick any. I just do total.
Yeah, metagenomics.
Oh,
are-
okay.
Yeah,
Okay. Yeah.
they'll get you there faster.
Yeah,
So you can see
okay.
what's happening.
Yeah, we
Some
could
of
do
it
that.
will mean that you're going to be including, unless you do the fractionation first, you're going to be including prophages. But
Yeah.
if you do the size fractionation first, do what
Yeah, that would be nice to do. Yes. There's no way of making a phage-free sample, is it? So we had some ideas at one point about trying to eliminate viruses and see what effect it has by kind of filtering a sediment extract and then using that as an inoculum. But do you think that's doomed to failure?
It won't work, but you can do, there's a pomegranate extract that will stop the phage.
Really?
Most of that, yeah.
A pomegranate.
What is that? Sorry, do you know how it stops it?
No, it's magic. But there's a magic pomegranate
So you can
extract.
toss add in, you know, EDTA
Really?
will stop.
And get rid
Yeah,
of
but
It's
it's
that.
not good for the
No, it didn't work.
bacteria.
It didn't work hard. Didn't want damage to the bacteria.
Mhm.
The EDTA. Okay.
So you're basically adding pomegranate extract, and that stops the phage.
Yeah, there's a Lipson. Look up David Lipson. He used
Uh-huh.
it in soils, and it worked pretty well.
So you would then make a Winogradzki column with pomegranate extract compared to one that didn't.
Exactly.
Okay.
Yeah, and I think you can denature it, so I think you can actually add the same.
Oh, I see. Yeah, that'd be
Yeah.
better. Yeah. Because that would be really nice, actually. The question would be like, do you get more diversity in the one with the phage?
So we made a synthetic gut one where it did, so in a mouse, where we took out the prophage, except for like one prophage.
Mm-hmm.
And then
Sorry, you didn't take out the propage.
we only had it, so the isolates were all prophage negative, except for one that we had in there. So you can do it that way too. So
Yeah, but then you've got to be able to
construct.
construct a synthetic system in the first place, and
Yeah.
we haven't really done that
But
as
you
yet.
should be able to do that. It should, all those things grow pretty nicely.
Yes,
All the pieces
Yes.
parts.
yes, it's true.
So if you think about your models on the one hand and the complexity of the
Yeah.
system on the other hand, it's quite a lot of work, of course,
Yes,
to get there. And are you thinking of trying to build also models in the experiments? Because if you can sterilize the system, you can also add like only 2 functional groups. Rather
yeah,
than
that's
the
what we
entire
that's what we
community.
want to do. Yes, that's what I meant by synthetic system, I guess. Or I mean, okay, so you could have a synthetic system that contains all the functional groups, or you could have one that, yeah,
Exactly. So you
Yeah.
can also build your
Yeah, that would be
experiments
very
in
nice.
the same way.
Yes.
As you want.
yes, yeah. We just need a new student for that.
But would you then get, in order to set up such systems, would you then get the inoculum from some prior experiments and analyze it and then say, okay, there.
Define strains, I think.
I think
That would
I
choose
think
for
you wouldn't do
defined-
defined strain. I think you would try and extract them from a well-developed white microcosm, actually.
You
Yeah,
can
this is what-
just
this is what
get
I-
like, get enough that you only have a handful of cells.
Yeah.
Is that gonna
Or it
work?
wouldn't. I mean, I would just take a ground-up microcosm community after 20 weeks or something, and then try to isolate from it cellulose degraders, and
Yeah, there were those, yeah.
because then you know that those ones actually do live in the microcosm environment. Yeah. But it's kind of like a 3-year project to do that. And
Yeah, it's really
yeah.
a couple of PhDs too.
Yeah.
Nice.
Okay.
I have a question. Can you check whether in different samples are exactly the same spectrum of species, or is there maybe in one log in Scotland there is a species which is not present in some other log? Because then, if there is a disparity of species, then of course, because there's no
Yeah.
relation of species, there can never be convergence.
I think you can never be sure that something's not present, because you can never sample ad infinitum. But you can certainly
It
say
could
that
be a
the
very small amount, which
yeah.
is undetectable.
yeah, you can say that. And now we've got this sequencing data, we can say much better about that, I think.
And then another question, because you say there's a positive selection for rare species, but maybe this is, can you really verify that all rare species are amplified? Or,
No,
or only
some of them are not.
Yeah, and then maybe.
Well, the ones
then
that didn't get amplified, you never see them.
I would not stress the result as an amplification of rare species, because it's just a new distribution of
Yes.
species. Then
Yeah.
then it's a necessity. As if there's redistribution of species.
Yeah,
then-
maybe it's fair
then
to
by
say that.
necessity, some of the rare species
Yeah.
get larger, and the larger
Yes.
get rarer.
Yeah, okay, that's probably true. But an interesting question would be the ones that came from rare, do they turn out to be somehow functionally relevant? And I think we should be able to tell that now we have this data. Yeah, okay. Should we take a little break? Before Jeff's talk? We've got 7 minutes.